Hi everyone, my name is Benjamin Risk, and I'm an associate professor here in the Department of Biostatistics and Bioinformatics and the Rollins School of Public Health at Emory University. And today I just wanted to share a little bit about some of the research I'm interested in. So let me see if I can share my screen here. Okay, so I have a website. If this sounds like stuff that's interesting, definitely check it out. And you can also email me if you have any questions. So the website's thebrisklab.org. If you can see that tiny little screen there. My first name's Benjamin, Ben, and my last name's Risk. So it's just the first initial last name. Um, and sometimes people call me Dr. Brisk, which is not my name, but that's fine if you call me that. Anyhow, so it's the Brisk Lab is what I call the students and myself and the collaborators I work with. And so yeah, here I have some details of the project. So one of the projects I'm working on in this plot here is looking at white matter development. So white matter is in the brain. It's the axonated the myelinated axons that are transmitting signals across the brain. And so one of the questions is how does the maturation of this white matter change across infancy? And so we're specifically interested in 0 to 6 months of age. And one of my collaborators, Dr. Sarah Schultz at the Marcus Autism Center, has collected data on infants. And this is something called a bivariate spline from a generalized additive mixed model. We have longitudinal data here. We account for the clustering, the correlation on repeated MRIs on the same kiddo. Or these are babies on the same babies. And we look at the effect of gestational age. So when the babies were born, 40 weeks is full term on this on this measure of white matter. This is called fractional anisotropy, and higher values mean uh, greater myelination, more white matter, more mature white matter here. And you can see that this is chronological age, days since birth, and there's an effective. So these are slightly premature. 40 weeks is full, so before 35 is premature, and then these 36 are not quite full term, but pretty close. And we're looking at what does that developmental trajectory look like as a function of both chronological age, days since birth, and gestational age. So that's one of the projects we're looking on. I have a little picture here you can see of this. So this is a spatial picture of what I was showing you before was the whole brain. And then this is looking at something called the voxel level. A voxel is a three-dimensional pixel. And the idea here is we looked at this measure of white matter, fractional anisotropy, at 30, 90, and 180 days since birth. Okay. And so these redder values mean greater white matter. It's a more mature white matter. And you can see how it does change a lot during these first 180 days. And really, what is just incredible here is the rate of change. So you look at this fractional anisotropy per day, and at 30 days, look how much faster it's changing than at 90 or at 180 days. So this is an exciting area of research where there's this thinking that these first 6 months, some people put the whole like 2 years. We're still trying to figure it out as a time of dramatic change in our brains and a time potentially that's really important to our long-term development. So things that happen in these first 6 months might actually impact us for the rest of our lives, but that's still an area of research. So I thought I'd show you another example of some of these brain images we're looking at. So this is a study that I'm also doing with the Marcus Autism Center. And we have children ages 8 to 13 that go into a scanner and they watch movies. And we collect eye tracking data on them. So whether or not they're blinking or fixating on the movies. So when you're paying attention to something, when you're really engaged in something, if you're listening to me right now on this video, then you're not blinking very much. You're just staring, eyes engaged, and paying attention to every word. But if you're not really interested in what I'm doing, you're actually blinking a lot, kind of looking around, and so we can measure attention using those eye blinks and those eye fixation events. So we categorize what your eye is doing as a fixation or an eye blink, and then we can create. This is a map of what happens in of the areas of the brain that are activated when a child is fixating on a movie. This is when they're blinking, and then this is what happens this is the contrast between eye fixation events when you're paying attention, and eye blinks when you're not paying attention. And it turns out this is a we didn't know what we were going to get when we started this project. We thought we were maybe going to get something called dorsal attention, which is when you're choosing what to pay attention to. This is actually more ventral attention, which means the children watching these movies are really doing so in a way that is more like, when you hear like an ambulance going by and your attention shifts to that ambulance, or when someone like calls your name and then you look and try to figure out what their name is. So that's like when you're reorienting your attention, and that seems to be the picture that we're getting in these children. These are all in neurotypical children. We also have some data on autistic children in the same age range, and so we're going to look at how these different attention areas of activation involved in attention change. So maybe I'll give you just a quick little more background on what we're looking at here. So I can kind of inflate these things. So that this is called a cortical surface. So I started this little presentation talking about white matter. That's not shown here. The white matter is actually underneath this cortical surface. So if I turned off all these images, this is actually the white matter surface. And then I'm overlaying this gray matter activation. So this is the neural bodies involved in the signal generation. And this is something that is captured. The activity there is captured using functional magnetic resonance imaging. So the cortex is this area of the brain that's 2 to 4 millimeters thick that wraps around your brain. And here, this is the temporal lobe. So this would be the front of um, your nose would be about here, your eyes would be about here, and so your ears are somewhere around here. And this area here is a temporal parietal junction, so that gets super activated. That's involved in attention and also like combining different senses. Like so, this also the children are listening, they're hearing a lot. So a lot of this is involved here is auditory. This area here is visual. So we got some interesting patterns of this what we're characterizing as the ventral attention network during this movie watching. This area here, this gray thing, is called the medial wall. So this is actually like the lateral side. This is you have you have a left hemisphere and a right hemisphere, and then in the middle, these are the medial areas here, and then there's actual space between your left and right hemisphere, but then you have this thing called the medial wall, where the cortex ends, and that's all your subcortical stuff going on down there. Anyhow, so I thought I'd just share a couple of those pictures that I was working on and looking at today, and I hope you find that interesting. And if you have any questions about Rollins or about neuroimaging, please email me at benjamin.risk@emory.edu. Thanks a lot.